हेलो नितिन. हेलो नितिन. हा नितीन आणखी खरा सर नाही जॉईन झाला. तर बघा खरा सर दिसतंय का?
नाही सर, नाही अजून जॉईन नाही झाले.
नाही अजून
नाही
सर.
ना. बरं ठीक आहे.
नाही सर.
मेसेज टाकतो सर.
हो सर.
हॅलो सर, गुड मॉर्निंग. हॅलो प्रोफेसर खरा सर. हॅलो सर, तुमचा आवाज येत नाही. आवाज येत नाही. वी कॅन्ट हिअर यू. शैलेश सर, माझा आवाज येतो?
येस सर.
येस सर, येतोय सर तुमचा.
Am I audible?
Yes, sir. Now
Hello?
you are audible. All right.
Am I audible?
Yes, sir. Yes.
I don't think I'm able to hear you.
Okay.
Am I audible?
Yes, sir. You are audible actually, but I don't know whether our voice can reach you or not. Am I audible to you? Hello.
Can you hear me?
Yes, sir. We can hear you, sir. We can hear you.
You are muted maybe or what? I don't
No. How about now?
able to hear you.
Oh, okay. What is happening then, Sneha?
Yes sir. Good morning all of you. I
नाही
welcome.
just a minute, Sneha. Because सरांना ऐकायला जात नाही आपण बोलतोय ते.
Yes. Okay. Yes, sir.
हॅलो.
Hello, sir.
I can't hear you.
You can't hear you. Okay. Just a minute, sir. Hello.
सर मला आवाज येत नाहीये.
नाही ऍक्च्युली तुम्ही जे बोलताय ते आम्हाला ऐकू येतोय, परंतु आमचा तुमच्याकडे जात नाहीये.
हो ते
नाही मला काय वाटतंय तो तुमचा हे जो काही की नाही काय म्हणतो त्याला
ब्लूटूथ.
राईट.
ब्लूटूथ. मी बघतोय, तुम्ही 1 मिनिट थांबा. मी बघतोय, डिस्कनेक्ट करून पुन्हा मला यातून बाहेर पडून पुन्हा एकदा लावावं लागेल, काय माहिती. हा ठीक आहे. ओके. Am I audible now?
Yes, sir. Yes, sir.
Ah, okay. Now
How
I
about we?
can hear you.
How about we? Are we audible?
Yes, yes, perfectly fine. Perfect.
That's perfectly fine. All right, sir. Okay, so then we will begin. Sneha, please go ahead.
Good morning, all of you. I welcome all of you on this online platform for the fourth lecture in this online lecture series on advances in life science research, ALSR 2022, under STRIDE Component 1 and Maharashtra Academy of Science, Kolhapur chapter, organized jointly by Department of Biochemistry and Microbiology of Shivaji University, Kolhapur. Now I request to Professor Dr. K.D. Sonone sir to give introduction about the lecture series as well as welcome and introduce today's speaker, Professor Dr. Arun Kharaj sir.
Okay, thank you. Thank you very much, Neha. Hello, good morning, one and all. So, as we know, the lecture series which we have started and it had been inaugurated by Honorable Vice Chancellor Professor D.T. Shirke sir on 8th February this month. And for this function, we had actually Professor Shoel from Jahangirnagar University, Bangladesh, who had inaugurated and delivered inaugural speech, first talk in this particular lecture series. So we had organized this particular lecture series just to give importance about the life science research, recent finding in life science, and actually cutting research areas, cutting research work that all the eminent scientists, eminent faculty members from different research institutes as well as universities, they are doing particular research in life science area. So actually, this is the fourth lecture that we have organized, and that too for that purpose we have Professor Arun Kharaj sir from School of Life Sciences, Laboratory of Applied Microbiology, JNU, Jawaharlal Nehru University, Delhi, New Delhi. So I am indeed very much happy to actually introduce Professor Kharaj sir. That basically he's the alumni of our university, Shivaji University Kolhapur. He had completed his masters in MSc Microbiology from YC Science Karad way back in 1993, and immediately after he had joined at Indian Institute of Science, Bangalore, for his PhD work. So, and afterwards, after completion of PhD successfully from IISc Bangalore, he had completed two postdocs, one from Institute of Grenoble, France, and then afterwards he was nearly 7 years at the prestigious Rockefeller University, New York, USA. So he is having vast areas of experience, particularly nearly 26 years of experience he's having in terms of teaching and research. So he has started his career in June 1993 as a scientific assistant at Department of Biological Sciences, Tata Institute of Fundamental Research, Mumbai. Then in 1994 till January 2001, he had joined as a lecturer at Appasaheb Garware College, Pune. Then January 2001 till January 2007, he had joined as a postdoctoral fellow at the prestigious Rockefeller University, USA. Hello? Sneha, what has happened?
Yes, sir. Audible. You are audible.
I am audible, but
Yes,
my
yes.
screen has gone.
Very clear.
But anyway, if I am audible, my screen has gone. I will just continue. And
We
actually
see
after
you.
completion, yes, sir. After completion of postdoc from Rockefeller University, he had joined in January 2007 as a reader, Department of Biotechnology, Sub-Campus Usmanabad, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad, Maharashtra, India. And then from August 2008 till September 2017, he had joined as a professor, Department of Biotechnology, again sub-campus Usmanabad, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. And from September 2017 till date, he has joined as one of the prestigious and reputed university in India, that is the Jawaharlal Nehru University, and that too in the School of Life Sciences as a full-fledged professor. So his areas of interest and areas of research specializations are particularly antibiotic resistance genes, host pathogen interaction, environmental microbiology, medicinal plant, antimicrobial and anti-cancer potential. And he has earned several awards, several recognition to his credit, particularly as actually junior research fellow, then senior research fellow, then travel grant by travel grant by Nobel laureate James G. Watson, that we know popularly as J.D. Watson, who had contributed or developed this particular model, DNA double molecular model, to present work in genetics of bacteria and their phage in August 1998 to the Cold Spring Harbor Laboratory, one of the prestigious laboratory in New York, USA. Then afterwards, researchers as a national fellowship of the French Ministry of Science, CNRS fellow, a postdoctoral research work at the University of Joseph Fourier, presently known as the University of Grenoble, Grenoble, France, in 2000. Then postdoctoral research fellow at the Raman Research Laboratory, Bangalore. Then academic director of the Dr. Babasaheb Ambedkar Marathwada University Subcampus, Usmanabad. Director, UGC Human Resource Development Center, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. And then again worked as a director at the same university from June 17 till September 17. So he has developed several international collaborations, actually particularly with France. Then again, participated as the previous in the multinational and international research project for the pneumococcal resistance in particular to understand about the actually with this virulence factors. He had published several international as well as national research papers and published peer-reviewed journals, books, and his contribution is very nice contribution for the particular development of microbiology and to the area of life sciences. So we have a very actually this eminent scientist, eminent faculty member who has got immense experience, very nice experience in academic as well as in research. So for this, welcome actually Professor Arun Kharat sir for this particular lecture series. And on the behalf of Shivaji University Kolhapur, Department of Biochemistry, Department of Microbiology, we welcome you wholeheartedly, and we request you to deliver your guest lecture in this particular lecture series. Thank you very much, sir. Good morning.
Thank you very much, Professor Sonawani, for a very kind, elaborate, profuse introduction. And it's my alma mater. Kolhapur is my alma mater. So I feel very privileged and very contented to deliver a talk on virtual conference basis. There are like several aspects which you did touch in the introduction about microbiology we did in our lab. And one of the major areas is antimicrobial resistance. So there are certain issues, forget about the global issues, but national level, there are certain issues which are not yet actually dealt in India about Indian side. And the biggest aspect is that India holds almost 20% of the global population residing in such a small, dense area. It's the hub for the pharmaceutical production of antibiotics. And also, the types of use of antibiotic will come to that when I make presentations. And roaring antibiotic resistance in India is unexplored at this moment. Meaning, what level of resistance is there, where it comes from, what it comes from is completely unknown. So there is something area called antimicrobial stewardship. So antimicrobial antibiotic stewardship in India is almost at a zero level. And that is very diplomatically, politely, I'm saying zero level, which I could say bluntly, it is almost zero. Rather than that, I could say it is a zero, but I don't want to say it because somebody will outburstly come and say that we have done this, we have done that. But whatever we have done in the light of developed countries and also lots of industrialized countries, India stands to be way behind. And before we come to the topic, I would come that you all have experienced or heard a death in COVID, which was among your kin relatives or the distantly related families. And much of the deaths, almost more than 60% of the COVID deaths in India, are in a very interesting age group, that is 30 to 59. Which is very difficult to explicate because globally, the senior adults have been mainly targeted. So why 30 to 59? And what is that? So it's because of one of the things is that inappropriate use of antibiotics. Inappropriate use of antibiotics. Because the student of microbiology would know that viruses cannot be treated with antibiotic. But you all know that the ICMR prescribed regimen also did contain azithromycin and ivermectin, which are the antibiotics. So not all the time antibiotics makes a miracle, but they are being used, and that is basically the overuse of antibiotic, which is called the drug abuse. So, despite having crores of people taking on paper the antibiotic, there is no prevalence rate either for ivermectin or for the azithromycin, neither before the COVID protocol nor after the COVID protocol. So it's very interesting and very appealing fact that I have presented before you. So what I'm right now, I also happen to be a academic editor of one of the journals, which is a very good journal, that Environmental and Public Health. So today I'll bring you, walk you through one of our project related work, that is how bad the environment today is with reference to antibiotics. And where all from, this slide basically shows where all from antibiotic gets into the environment. And it would not be surprising that anything that you eat at home, whether it is a vegetable or whether it is a non-vegetable, no matter how much you wash, 2 times, 3 times, lukewarm water, plain water, running water, you are consuming antibiotic resistant genes. You are eating all the organisms. And you would say that we cook. Hey, but when you cook, you are basically getting into the dissociation reassociation kinetics. You are not getting rid of the genes, you are not getting rid of the DNA, and you are giving away for natural transfer or genetic recombinations in your body. You are sponsoring your body as a bioreactor for cloning and developing all sorts of resistant derivatives. And then again, so you can see the aquaculture can't be considered without the use of chemicals and their effluents, which will go into the wastewater treatment plants. They are also full of chemicals, and some of the chemicals are antibiotic. Then the farm animals, as you all know, that their feed is no longer open feed. In the farm animal, the feeding material contains the antimicrobials already, just to be on the preventive note, not to get them into the dreadful diseases, not to get them ill, sick, but that what is so-called prophylaxis is too much of prophylaxis. But then they will release the urine, they will release the feces, all that goes run off into the aquatic sources, whether it is the surface water, whether it is any another water or groundwater that goes into. And the whole story started in Usmanabad when I was doing my dispersing my duties in Usmanabad as a reader. I realized very surprisingly, Usmanabad has 95% of water consumption coming from the tube-well water. And 95% people have had, interestingly, Pseudomonas aeruginosa in their tube-well water. Whether I take it from the house, whether I take it from the domestic disposal, and whether I take it from anywhere. So that was a very interesting that the water has Pseudomonas aeruginosa, and that to 300 to 400 feet underground. So that was very crazy. That was a very weird observation, and that's where I initially started doing antibiotics. Otherwise, I was not at all interested in antibiotics. And then we started to see that initially we were doing phenotypic, then we started doing genotypic, and now we do all molecular levels. And we know that large number of resistant bugs are there, and that you can't perceive on the basis of perception that at 300, 400 feet underground, there is antibiotic. There is no direct selection as such. But this water, this material percolates through the capillaries way down there, and organisms get a trace very low amount of concentrations of these antibiotics, serves themselves as a selection pressure, and these resistances as a result of natural selection. Then agricultural soils are also full with chemicals, fertilizers, and antimicrobials and pesticides, etc. You are unknowingly consuming pesticides, unknowingly, no matter how much you want to go away, remember all pesticides are hydrophobic in nature. They don't easily get solubilized in the water, and therefore it would not be very wrong to say that. And their boiling melting temperatures are well above your normal temperature. So obviously, the most of those pesticides are consumed by us, no matter how many times we wash it off, how many times we fill it off. And through the slush, through the runoff water, whether it is from the irrigation, whether the food that we eat, as I said, will be the source of domestic use, food material, sewage, everywhere we are consuming antibiotic resistant bacteria, antibiotic resistant fungi, and then we are walking all over. We handle all these things. So from different sources to human and from human to different sources, and the cycle continues. And that's how the environment has become day by day worst with reference to antimicrobial resistant bugs. So now these are the bacteria, as we all know, E. coli, which is considered to be a paradigm, and the quickly reproducing bacteria. It is said to be 30 minutes, but I know lots of strains who multiply within 12 to 18 minutes also. But this is an average time. Now, how quickly the genetic transfer can occur? One of the things is that sex in bacteria. Bacteria do exchange genes and form a new combination. Bacteria exchange genes by means of course, a transformation, naked DNA, transduction through viruses, that is bacteriophage, and also the sexual mechanism, that's conjugation. And this involves a transfer of genetic material unilaterally via a cytoplasmic bridge produced between the donor cell and the recipient cell. Now, if you do look at the text of microbiology, typical text of microbiology, and the original experiment conducted by the Cavalli and many people, Liederberg probably you would know, but HFR was devised by the Cavalli. So when you look at the Cavalli's experiment, you would see that, or S. Benzer's work, you can see that. The E. coli K12 and E. coli B, these were the different strains of E. coli, but they could not engage conjugation. That means B will engage with B, C will engage with C, K12 will engage with K12. That was the basic fundamental principle. And it was too difficult to demonstrate conjugation experiment in the lab. Average laboratories have been demonstrating or conducting the practicals for plasmid transformation, and the transduction. But conjugation has been always a pain in the neck situation. And it was very difficult. But hey, when you come into the nature, forget about E. coli K12 conjugating with E. coli B, but unrelated species of bacteria. What I mean to say that Salmonella engaging with Pseudomonas, which don't belong to the same family, meaning very dissimilar organisms have been able to conduct conjugation, and originally the conjugation was between the two strains of the same species or same cultivar, and that whole transformation, whole paradigm shift in the process of conjugation has made huge impact on the word called evolution. Evolution was convergent evolution or a divergent evolution. And you all know that when you isolate a bacterium, what you do? You will do the 16S sequencing. And that is to know why 16S, because telltale story is that it is highly conserved and there are certain pockets which are variable. And if you do see the variations in that variable regions, you can attribute those changes to the species differentiation. That's very simple definition. Otherwise, if you see a base change in the conserved domain, it will trace it back to thousands of years or millions of year divergence of the species from one species to the other species. That was very strong wording and definition of molecular clock. But this conjugation business, when it moved, forget about the two strains of the species, interspecies, intergenus, and interfamily, and all together unrelated organism. Now realize that the evolution can occur within a night. Why are we talking about millions of years tracing back the evolution? Evolution is happening within a night because of the natural selection. If there is antibiotic, there is a condition called do or die. Let's just think about somebody who barely knows the swimming, has jumped into the water, whether deep, will try to come out of it. Everybody, even if somebody who sounds, has tried to survive, right? So same is true here also, that they would like to cope up with the antibiotic concentration in the environment, and the best method is to acquire the genetic material if there exists in the nature surrounding in the niche. And that's where they forgotten all the limitations of E. coli, Quetel, E. coli, B, etc., and unrelated species have engaged. Now, this has brought a new term called horizontal gene transfer and horizontal evolution. So, vertical evolution still remains very difficult, but horizontal evolution is much rapid, much a faster process. Recent studies on bacteria in the world wild have shown that it occurs in the soil, it occurs in the fresh water, it occurs in the ocean, and it occurs inside of the living organism. I already said that we sponsor our body. It was treated antibiotic as a revolutionized medicine back in 1929, which was discovered by the Alexander Fleming, and which was later purified by the Florian chain, was extensively used before purification in the Second World War to treat the wounds of the soldiers. And after that, many more type of antibiotics have been developed. Today, we have more than hundreds of types of antibiotic in use. Most are inhibitors of either protein synthesis, blocking 70S ribosome, large subunit or a small subunit, which is the typical property of bacteria. Remaining are affecting either bacterial cytoskeleton, that could be the cell wall, that could be the DNA synthesis, RNA synthesis, cell membrane, or other metabolic steps. Now, when it was generously applied in 1940 or 41, it took only 20 years or around 20 years to show the first antibiotic resistance. First antibiotic resistance was seen in around 1961 in United Kingdom. That was a Staphylococcus aureus, which was able to show the antibiotic resistance in the bacteremia, and pneumonia started to show resistance. Now, if you really go back to the plasmid classification, the R plasmids were identified and demonstrated, detected in Shigella by scientist Shiga well before 1960. And that was a multiple drug resistance term. The term wasn't used, but it had at least the R factor, the first R factor of the world, had 8 different antibiotics resistant properties. Today we can call it like genes for the 8 different antibiotic resistance were located on the first R factor. And today there are various different strains of Staphylococcus aureus whose resistance is growing away from antibiotic use. Terms such as the methicillin resistant Staphylococcus aureus or vancomycin intermediate Staphylococcus aureus, vancomycin resistant Staphylococcus aureus are becoming the major issues of the time. Now, multiple resistance. Some of the resistance was already there in the nature, in the bacterial population. The presence of antibiotic in the environment in higher concentration increase the pressure, and thereby the bacteria are forced to acquire the resistance, whether in the human body or in the environment. Now, resistant bacteria which survived to the natural selection have got very rapid replication, and that is a selective advantage, and automatically the sensitive population is diluted out. Then they pass their resistant genes onto the other bacteria, and regardless whether the bacteria causes a disease or bacteria doesn't cause a disease. So dissemination of genes can also be because of the transposons, can also be because of integrons, and this has been shown in 1958 by the Shigella flexneri strains. Now, antibiotics basically promote the resistance, and you can see that if a patient is taking a course of antibiotic or patient is prescribed a course of antibiotic who does not complete it, It could be because the financial reasons, or another very common reason is that अब मुझे ठीक लग रहा है, मुझे problem नहीं लगता है, so I'm not going to take medication. Or somebody who forgets to take a dose on regular basis. Automatically it gets a resistance to develop. And antibiotics also kill then other innocent organisms which are actually useful, which are non-pathogenic, meaning both saprophytic and useful microbes will be killed by the antibiotics. That may include namely your gut flora, and gut flora is so required to maintain the health of your intestine. And because of antibiotics, आपने देखा होगा कि एंटीबायोटिक थेरेपी की है किसी को, और एंटीबायोटिक थेरेपी के बाद उसको एंटीबायोटिक इंड्यूस्ड डायरिया हुआ. कभी देखा है आपने किसी ने? कोई अनम्यूट करके बताएं. एंटीबायोटिक इंड्यूस्ड डायरिया कभी पेशेंट को हॉस्पिटल में होते देखा है? Has anybody seen? A patient was put up in the hospital for 2 days, he or she was okay, getting better. और तिसऱ्या चौथ्या दिवसापासून त्याला डायरिया सुरू झाला. असं झालं कधी? पर्टिक्युलरली लहान मुलांना, infants. Or you cannot unmute to tell me. Have you seen ever a patient who was hospitalized, felt better, but after 2-3 days, the child started to get diarrhea in the hospital? No? Nobody says anything. Anyways, but that happens in the children's particularly. And that's because of the normal flora gets washed off as a result of antibiotic therapy. And now the intestine is not as healthy as supposed to be. Now, probiotic is a need at that time, which will reduce the competition for the resistance pathogens. The use of antibiotics also promotes antibiotic resistance in the other organisms. These non-pathogens then later on pass the resistant genes to the pathogenic organism. Now resistance gets from one person to the other person, and number of antibiotic resistance bacteria increases in the family members also. In the places where antibiotics are used extensively, such as the farms, poultry, then agricultural field, hospitals, and that's the places where a large amount of resistance can be developed. Now, viral infections, I already told that they cannot be treated with antibiotics. However, doctor continues to prescribe the antibiotic, and they are being used as a prophylactic measure, but the prophylactic measure itself is also an abuse of antibiotic. Self-medication, lot of time, an educated population is believing that चलो medical store पे चले जाते हैं, उसे हम कहेंगे कि basically pharmacists and pharmacists who might be 10th class, 12th class will prescribe you antibiotic. Empirical treatment and blanket therapy. These are the terms. Like you are going to the hospital, doctor is asking you the same signs and the symptoms, and based on his experience, without microbial culture or antibiotic sensitivity test, doctor is prescribing you an antibiotic. Or blanket therapy, when you know nothing, you simply give one cell wall inhibitor, one protein synthesis inhibitor, one DNA synthesis inhibitor, and that is called a blanket therapy. Or treatment in general without a clinical microbiology. That is what happens in India almost 98% times. Now, the beta lactams are known to be resisted by the enzyme called beta lactamase. And as you could see that the resistance is growing as the time passes. So you can see that the certain traits such as BLA-TAME and BLA-SHV are growing day by day. Similarly, TATM, TATO, TATQ, and TATW are the antibiotic resistant genes which are increasing in the nature. For erythromycin, ERMB, ERMC, ERME, and ERMF are increasing in the nature, especially ERMC. Or in the case of TATQ is on rise actually. Now, when we talk about the animals, the horses, the rabbit, the fish, the sheep, the poultry, the pig, and the cattle. So you can see the highest resistance is among the cattle and the horses because these are the ones. and the third comes the poultry. So these are the ones which are used for grazing, milking, and for eatings also. So other examples, basically here, types of antibiotic resistance, you can see tetracycline is on the number one, then come the group of penicillin, then come the sulfonamide, and all other classes of antibiotic resistance is also reported throughout. Popovus et al. Has shown it already. Okay, so you can see the which organisms have been shown: Salmonella typhi, E. coli, Klebsiella, other members of Enterobacteriaceae, Enterococcus species, then Staphylococcus, all are known to be considering from the, and where was it demonstrated? In poultry, in the egg cell, in the egg yolk, in the liver of the animal, in the water of the environmental dust, etc. People have shown. People have shown it from the Middle East. People have shown it from the Southeast Asia. People have shown it from the Europe. And people have shown it from the United Kingdom. But unfortunately, no Indian citation is here. So Canada, US, then again China to some extent, France, Lebanon, South Africa, they all have shown number of different antibiotic resistant genes in various pathogens of gram-negative, especially E. coli and Salmonella chhod do, so something like hospital community acquired infectious agents or nosocomial. So even in northwest part of the Europe, Western Europe, most of the countries have documented, but you will see that none of them have India. So you can see the highest again, if we talk about animal production worldwide, in that poultry is on the number one, the pigs are on the second, and the cattle are on the third. So mostly the resistance is because the current antimicrobial resistance is because the chronic infections which have a poor adherence to the antibiotic, agricultural use, and empirical therapy by the doctors in the developing world. And basically, antibiotic then causes either target modification or enzymatic degradation, which is very well known in the beta lactams, cephalosporins, and the penicillin group of them. Abuse, basically, antibiotics are treated as a prophylactic measure for viral treatment or viral infections. Now that causes increased egg flux, the decreased egg flux, which is commonly found with the vancomycin and tetracycline group of organisms. Overuse of antibiotic, you can see the COVID-19 treatment. Antibiotic-based sanitizers have been made. आपने advertise में ये भी सुना होगा कि these days paints are coming with antimicrobial. So just think that where all people are using antibiotic containing mixed paints are being used. So to make sure that आपकी wall में fungus नहीं लगेगा. Now that would cause a membrane depolarization and persistent cell information, and that puts the antimicrobial resistance at a top position as a paramount issue. Now antibiotics are disinfectant. If you do look at it, susceptible population was very high to begin. Then later on it came the basically the susceptible, some resistance, and dead cells. Then dead cells were high in number. Among that, a few cells started to become resistant, and then today the resistant bacteria are increased so much, and persistent cells are getting back into the recycling manner, and shows that you are full with the antimicrobial resistance. There are certain examples of penamis, carbapenems, then cephalosporins, then DNA synthesis inhibitors. All different kind of antibiotics have shown to have a significant amount of resistance. Now, the total upper daily dose of common antibiotic compared to the drugs from the 2013, it has been shown that the vancomycin, meropenem, ceftazidime, augmentin, the cephalexin, and metronidazole use has, or ciprofloxacin use has been increased too much high in the antibacterial cases. If you look at a diabetes, if you look at antivirals, if you look at a metabolic diseases or the central nervous system or coronary vascular diseases, the resistance or the utilities are very low, but the antibiotic utility is too high in amount, and that is what is also the causing the natural pressure. So basically, what happens now? These antibiotics are basically controlling the resistance emergence in the environment through the animal, stock animal, grazing animal, and the aquaculture. Now, these antimicrobials will get into the domestic and hospital system. Now, hospital also generates the antimicrobials, antibiotic resistant bugs, and they are being sent into the environment, in the nearby environment, and that is establishing the comprehensive data on the antibiotic resistant genes in the data analysis these days. So you can see that minimizing the spread of antibiotic resistance in the environment is a need of ours. Now, this is our project that we have been endeavoring recently. Now, studies on identifying the financially feasible policies to reduce the load of antibiotic resistance in the environment. So this is a project sanctioned to us by the ICSSR. Now, basically, the one of the aspect that is terminal aspect is the questionnaire, and the rest of the aspects were experiment based. There are no slides made for the experiment based, but we have selected 4 locations. One is absolutely Delhi. Second is Bareilly. Now, these are the 2 locations where you have the cultivation of the vegetables just on the bank of river, and these are the polluted river. Then the third one is from the Varanasi, where the religious and anthropological activities do take place, and there also we look antibiotic resistance in the vegetables, leafy vegetables particularly, the antibiotic resistance. And the fourth is the Panchaprayag, that is Alaknanda river in Vishnu Prayag, has the Dhauli Ganga and Alaknanda union, that is the Sangam. Then in the Nand Prayag, Nandakini and Alaknanda confluence. Then third one is in Karna Prayag, where Pindari and Alaknanda confluence. The fourth Prayag is Rudra Prayag, where the Mandakini and Alaknanda confluence. And then the fifth Prayag is Dev Prayag, where the actually Ganga starts from there. So it's Alaknanda and the other river, the fifth one. From there we come to the Rishikesh, where actually the anthropological contamination activity starts because tourism. And from there we come up to Haridwar. That's where the religious activity and industrialization on the bank of Ganga starts. So I start with the Vishnu Prayag and then come all the way up to the Haridwar. That comes up to the 5 Prayag, 5 confluences and 2 sites. One is purely anthropological and 7th one is anthropological and as well as industrialization and agriculture. And we find that nearly when you come to Rishikesh and Haridwar, a huge number of enterobacterial number goes on rise because of the human interventions and industrial. And in Haridwar, we see lots of antibiotic resistant bacteria. While in the case of Okla, which is a site in the Delhi, almost every vegetable that we have selected, there are 4 vegetables, have got 90% bacteria, enteric bacteria isolated, whether they are in the leaf, on the leaf, that is a phylospheric, or mycorrhiza. They all have multiple drug resistance status. And same is the story with the Bareilly site and as well as Varanasi site. So that was one of the aspect of experiment. The slides are not. But the second aspect is about to what extent the society knows about antibiotic uses. So the questionnaire was made, and to assess the knowledge of antibiotic and antibiotic resistance at community level, data was collected from 5 different categories. So the senior secondary school student, their parents and their teachers. Then senior college and university level students and the teachers. Then the medical, paramedical, healthcare workers who are working directly, indirectly into the hospital setups. Then the learned groups from the corporate sector, so they're mostly from the national and multinational companies, as well as the non-government organizations who actually work for the environmental sector. So we were collecting the information, and what we find, done the statistical analysis, and we found that out of 1,000 different response, our questionnaire was divided like to what level they know the antibiotic. Do they know about antibiotic resistance? Do they know about use and abuse of antibiotic resistance? What is their perception about action to take to reduce the antibiotic resistance? Their attitude towards the medical practice and general medicine. Those were the things. So we find that largely most 96% people knew a little bit, they had heard about antibiotics, and most of the information they got was from the news and the books. Now these books are not necessarily coming from the textbooks, these could be any type of sources, they would know it. And group discussion, common parlance, all those things were like almost 40% of them, and they were able to differentiate or not to able to differentiate antibiotic and other drugs. You could see that almost 60% people were able to differentiate, and rest of them did not know how to differentiate antibiotic from the any other drugs. And interestingly, almost up to 90% of the people said that it was medical or paramedical personnel who prescribed the antibiotic for them. While for the remaining population, it was almost a 10% population was basically the other group of it. Now, antibiotics are required for the treating the disease. A perception says that most of the people agree that antibiotic is so required for the treatment of the disease. Now, we had internal controls also introduced, in which we find that uh, we know that antibiotics can't cure the viral diseases. Antibiotic can't cure the general terms. So, for example, fever, cold, flu, HIV/AIDS, malaria, measles, these were the headaches, body aches, and COVID-19. We for sure know that these infections, these conditions cannot be treated with antibiotic, but you can see that surprisingly, 13% people believe that antibiotics can kill or treat the HIV. 70% people believe that fever and 40% headache can be, 40% people say headache can be treated with antibiotic. 50% people say malaria can be treated with antibiotic. 35% people say that measles can be treated with antibiotic. 40% people believe that COVID can be treated with antibiotics. So it was sufficient to see that though they have heard about antibiotics, though they get antibiotic in use, but they really did not know where to use antibiotic, for what purpose they should be using the antibiotic. Now, antibiotic resistance term was heard almost by 80%. Antimicrobial resistance was a bit confusing term, oxymoronic situation, only 50%. MDR, extensively drug resistance, pan drug, and superbugs, they were randomly like 60%, 40%. You can see that their understanding was pretty weak. Now, use of antibiotic prescribed by the doctor was the largest, and the repeated use of antibiotic was also experienced by the higher education people. And antibiotic without prescription, people said that almost 40% people were able to do that. In the case of higher education, when we look at antibiotic, a lot of people, 30% people, did not even remember when antibiotic was prescribed, and believe they can save, antibiotic can save every time they are sick, or sort of that question. We realized that there was also strong agreement was only there for the 30% people, while rest of the people did not agree that always antibiotic can save your life. So that was good. And under the subteam, we found that improper use of antibiotic can lead to the ineffective treatment was agreed by the 50%. Again, 60% people thought that increases in intensity of the illness. Emergence of bacterial resistance was seen by the 60%, and additional burden to the medical cost was experienced, said by the 65% people. Now, we know how it works, was said by 30%. Another important thing that disease symptoms were not as good. So you can see that use and misuse of antibiotic can also be seen, that pharmacists and medical store people usually advise people. Then action towards antibiotic, people say that people should not use antibiotic when it is not required, when doctors are prescribing, etc. All those kinds of studies were carried out to be able to estimate their action towards antibiotic. Okay, so the resistance can be developed due to the skipping of the dose, or treating that antibiotics are safe and therefore one can commonly use it. So we went to the too much in details of all these kinds of questions, and we realized that most of the people visit their doctor, almost 50% people visit their family doctor, 50% population visits randomly. So it's a bit of a chaos. And how did you stop your antibiotic course? 35% people believe that when we felt better, and only 55% people believe that we have completely followed the prescribed regimen by the doctor. So you can see that there is also a great amount of problem. Additional educational legal response, we have checked again into the different, I already mentioned about that to you. And now I will stop here to take any more questions if you have. Okay, I'll be taking the question.
Hello, students. Are there any questions? Topic is open for discussion. Please go ahead if you have any questions, any doubts. Hello, faculty members, students. I think there are no any questions.
Yes.
Yes.
Thank you very much for inviting and having me here.
Yeah, exactly. Okay, Sneha, go ahead.
Thank you, sir, for such an informative talk on a global threat of antibiotic resistance. Now I request Dr. S.R. Wagmare, sir, for vote of thanks.
Thank you, Sneha. Am I audible?
Yes, sir. Yes, sir.
Okay. So as we know that today is the fourth session of our online lecture series that we have started on the 8th February. And on behalf of the Department of Microbiology, it gives me an immense pleasure to give the vote of thanks of this session, which is under the online lecture series organized as an Advances in Life Science Research under the UGC-STRIDE and Maharashtra Academy of Sciences, Kolhapur chapter. Respected Sonone sir, today's eminent personality, that is Professor Arun Kharat sir, all faculty members, dear students. As Arun Kharat said, very well pointed out that whatever the antibiotics we are getting today, that I must to say that now when you are using the antibiotics, we have to to keep it in your mind that there should be one slogan that handle with care, because by 2050 antibiotic resistance would be the biggest threat for the whole world. So if we are not getting to aware about it, because sir has very well pointed out that what is the antibiotic, if you are not using that antibiotic properly, what will leads to it. And as we know that in our Department of Microbiology, under the the guidance of Sonare sir, there is a lot of research is also going on the antibiotic resistance. Kharat sir is also have been focused for so many years that what is the cause of antibiotic resistance, and that gives the breakthrough for to control the such antibiotic resistance pathogen. So I'm very much thankful to the Kharat sir for awareing this antibiotic resistance problem to the current youth, because they will be the future and they must to know that how to use the antibiotics. So I'm very much thankful, sir, for delivering such a nice lecture, knowledgeable lecture to our students. I'm also thankful to the Sonone sir, all the respected faculty members, and all the dear students. Thank you, one and all.
Okay, thank you, sir.
Thank you, sir. Thank you. Thank you. Thank you.
Yes, it's very nice. It was indeed very nice to have you here on this particular platform.
Thank you, sir.
Yes,
Thank you.
thank you very much, sir. Thank you very much. Good day.